Yeah, thank you, Tatiana, for the great introduction. And thank you to the Bayer Foundation for giving me the opportunity to present some of the work that Bayer has been doing in genome editing. And I'm also happy to share the stage with an accomplished young academic such as Simon, who's after me, going to present some really very innovative ways of applying genome editing to cardiovascular diseases. I'm assuming you can share my screen. You can see my screen. If not, yep, thank you. Awesome. So for these boundary breaking science lectures, it's customary that the panelists or the presenters will briefly introduce themselves. So as Tatiana's already mentioned, I'm Florian Richter. Currently, I'm a scientist at the Bayer Gene Editing Group. A brief CV is on this slide. So I started doing regular biochemistry as an undergrad in Zurich, where this was before genome editing was known. So back then focused on enzyme engineering and molecular modeling. Then for my PhD in 2006, went to the University of Washington in Seattle and did sort of the early computational enzyme design efforts with David Baker. Then after the PhD, I moved back to Europe and did a postdoc in Berlin, where I already started working on Cas9 and tried to engineer light switchable variants of Cas9. That was together with Andreas Möglich, and also spent some time in Emmanuel Charpentier's lab. And finally, in 2016, went to the most beautiful city that's on this slide, namely to Wuppertal, to start working at Bayer. And so since starting to work at Bayer, I've developed or helped develop the gene editing portfolio and also done some enzyme engineering. So today, I'd like to first give an overview of current gene editing methods and how they can be clinically applied. And then I can also share a few things about Bayer's emerging portfolio in the gene editing space. So let's get started. I think most people that are currently in the session have heard of Cas9, but I think it helps to, again, just focus on the basics of Cas9. So what is Cas9? Cas9 is essentially an enzyme that binds to a guide RNA, and if the guide RNA has the right sequence and a piece of DNA with the same sequence is around, then that a protein RNA complex will bind to the DNA sequence and cleave it. Relatively simple. And so since the RNA can be freely programmed, Cas9 can be reprogrammed to target any DNA sequence. And so that means any location in the genome can be easily targeted by simply providing the right guide RNA. And this really trivial programmability of CRISPR-Cas of Cas9 is what essentially allowed the genome editing revolution. Because now essentially everybody can design a reagent that can specifically target a certain location in the genome. Now, where does such an amazing molecule come from? So I'd also like to briefly share essentially the foundational science that this CRISPR-Cas genome editing revolution rests on. So Cas9 has been around for a long time, for probably hundreds of millions of years, and originally evolved as part of a bacterial immune system to defend bacteria against phages. And the way this CRISPR mechanism in a nutshell works is that once a phage or a bacterial virus invades the bacterial cells, parts of the DNA of this virus are being integrated into the bacterial genome, and then you have a locus in the bacterial genome that has bits and pieces of different viral DNAs, and then from this locus of viral DNAs, the guide RNAs are being formed. Cas9 is loaded with the guide RNA, and then the next time the virus invades the bacteria is ready to defend against it. And what's also important to understand about this is that these CRISPR-Cas systems are essentially the result of molecular arms race for the last probably hundreds of millions of years. And while Cas9 is the most famous or most known RNA programmable nuclease, many diverse subfamilies exist. And so nowadays they're broadly divided into the Cas9 and Cas12 subtypes. And I'll briefly mention Cas12 again later when we're talking about what Bayer is doing in the space. Now that we have this molecule that can cleave DNA, the question is, how do you leverage this DNA cleavage activity to get a therapeutic of phenotypic effect? Because just cutting DNA isn't going to help you, right? That's essentially usually probably will just cause problems for the cell. But the key to understand this is to look at what's happening after the DNA is cleaved. And essentially, two different things can happen after the DNA is cleaved. So what happens most commonly is the cell will detect the double-strand break and then just stitch the double-strand break back together. And because the double-strand break isn't being repaired perfectly, you get small insertions or deletions. That's commonly termed an indel. And that usually means that the gene has gotten knocked out because you'll have introduced a frameshift mutation. And so the most common outcome of Cas9-based gene editing is disrupting the targeted gene, at least of native. Then some cells, if donor DNA is provided, can also integrate this donor DNA, and that's called homology-directed repair. And that's a way of making specific changes to the genome or precise modifications to the genome. But that really depends on the cell type. So that usually doesn't work in post-mitotic tissue. So take-home message is that with basic Cas9 or Cas9, the natural molecule, knocking out specific genes has become really trivial, but precise modifications of the genome are difficult to achieve with native Cas9. Now, because humans tend to be ingenious, after the discovery of essentially native Cas9, various people across the world have figured out beautiful adaptations or beautiful variants of it. And what is also key to understand is that Cas9 can be deactivated. So essentially there's two nuclease sites that can be deactivated with single point mutations on the amino acid sequence. And once you have deactivated these nucleosides, you have what is referred to a dead Cas9 or deactivated Cas9 or dCas9. And this protein can still bind DNA. You can also deactivate just a single of the active sites, and then you have a nickase Cas9 that will only cleave one of the strands. And now, as a next step in the sort of 2.0 approach, once you've deactivated Cas9 and have a programmable binding complex, you can attach other proteins to Cas9 by simply making fusion constructs, where again Cas9 is attached to another protein, where other proteins have different activities, and then Cas9 serves as a targeting or as a platform that can bring other activities or other enzymes to certain regions in the genome to then have their specific effect in the genome. A bunch of very creative things have been done in this regard, and therapeutically, some of the most interesting ones, and also the ones that Simon will mention in his talk, are methods for introducing specific point mutations, and these are called base editing and prime editing. And so I'll briefly introduce these two CRISPR 2.0 approaches. So something that's called base editing that I think Simon will talk about is a technique in which specific point mutations can be introduced. So there are certain enzymes that are called deaminases that will essentially perform chemistry on the DNA. So that will take a nucleoside or the base of the nucleoside and deaminate the amine group, and then you get an oxygen here. And what that means in terms of the DNA code is that a C can be turned into a T, and an A can be turned into a G. And in this way, base editors can install specific point mutations without cleaving the DNA. And so they're doing much more than just creating double-strand breaks. Now, these don't exist in nature. They're artificial constructs that were mainly engineered in David Liu's lab. And second example that Simon might also talk about is another 2.0 technique to introduce specific changes in the genome, and that's called prime editing, which is even more powerful. And that is if another enzyme that's called a reverse transcriptase is fused to Cas9, then upon Cas9 cleaving one of the DNA strands, this reverse transcriptase can use the cleaved DNA strand and use it essentially as a primer for an extension that has been added to the guide RNA. And then the reverse transcriptase will essentially back translate what is on the guide RNA into the genome. And so essentially anything that was programmed to be on the guide RNA will end up being written into the genome. So essentially prime editing is a way of writing artificial new sequences into the genome. And so it can be used to essentially introduce any single mutation and also small specific insertions or deletions into the genome. And this is also an artificial construct that was engineered by the same lab as base editing. Now that we have this sort of very nice portfolio of tools, so knocking out genes has become trivial, and with base editing and prime editing, making specific changes in the genome has also become possible. Question is, how do you apply these molecular tools to therapeutics? And when talking about therapeutic gene editing, there's essentially two distinctions, and that's you have ex vivo editing on the one side and in vivo editing on the other side. Ex vivo describes a process where one would take patients from a cell, then edit them in the lab in a dish, and you take the edited cells and inject them back into the patient. And these ex vivo approaches allow really precise control over the editing outcome, right? Because you will only put cells back into the patient that where you've done quality control on, and that have the edit that you want. But of course, it's limited to tissues where the edited cells will propagate, right? For example, in blood, this approach is really is very feasible. And since about 2 years ago, we have the first approved CRISPR treatment. That's Casgevy, which is a functional cure for sickle cell disease. That is an example for essentially ex vivo gene editing. And coincidentally, because essentially with Cas9, because you can reprogram Cas9, it's relatively easy to, again, target certain loci. And so since targeting certain loci has become really easy in academic settings, it was nice to see that this is also made clinical development faster than what was often possible before, right? So the Casgevy clinical development pipeline was about 5 years between first patient being dosed and approval in 2024. Now, perhaps even more powerful is the in vivo gene editing approach, where you would take the gene editing reagents, and that is also what Simon will be talking about, where you take the gene editing reagents in some vector, either a viral vector or a lipid nanoparticle or something, and then the editing happens inside a patient's body. Now, this in vivo editing is dependent on the availability of a delivery technology, and that is currently still the biggest roadblock in translating G into the clinic. So while certain tissues like liver are really easily targetable at the moment, other tissues are still somewhat difficult to target. But there are recent also advances in targeting cardiovascular tissue, which again, Simon will mention. Now, when talking about therapeutic genome editing, I think can't do without mentioning the baby KJ story, because it's really one of the most inspiring and innovative things to have happened in the medicine space, I would say, in the last couple of years. So last year there was a case that was published about an infant patient that had a severe metabolic disorder that presented right after birth and led to a severe worsening of health right after birth. And upon sequencing the patient's genome, it was discovered that the patient had a mutation in a urea cycle protein. And then within only 7 months, a specific personalized mutation correction strategy that was based on base editing was developed for this patient. And I think the patient received 2 doses, or I think by now 3 or 4, of this specifically developed gene editing reagents, and now child is apparently cured. So while this case really shows the potential of in vitro gene edit or in vivo gene editing, and essentially the aspiration and the hope, however, it's important to mention that it's also still an exception, because the success in this case was enabled by the availability of a suitable base editor, and also the fact that the disease manifested in a tissue that was inherently targetable, and the liver in this case. Now, finishing on the introduction to gene editing, I'd like to finish with a very brief overview of essentially the progress of CRISPR-Cas from first essentially academic paper showing that Cas9 can be used to specifically leave DNA, which happened in 2012, to essentially the first approved therapy in 2024, and then this recent in vivo editing breakthrough. And it's really a spectacularly short timeline when you think about biotech, right? From something that's being discovered, and then only 10 or 11 years later, you have essentially real clinical benefits. There's a bit of caveat, of course. Cas9 wasn't only discovered in 2012, right? A lot of foundational science happened before, in the 10 to 20 years before. But I think, again, Cas9 revolution started in 2012, and then within 12 years now, there's multiple clinical trials and even things on the market. Now, transitioning to the second part of the talk, on the bottom of the timeline, I've indicated Bayer's activities in the gene editing and CRISPR field. And so over the years, Bayer has done multiple collaborations. with different gene editing companies, but has also done some internal activities. And so two examples are that we've started internal nuclease discovery and characterization workflows, and I have also started exploring gene editing indications ourselves, which will be the next section of the talk. So I will give a brief introduction to our emerging gene editing technology portfolio before then handing off to Simon. So one thing that's important to understand about Bayer, and Tatiana, you mentioned there's also guests from St. Louis. So Bayer isn't just a pharma company, right? But Bayer is also a large crop sciences company. And most crop sciences companies, or many crop science companies, have over the years or decades collected large strains of internal microbial databases, or large strain collections of bacteria that grow in diverse environments, most importantly agricultural ecosystems, but also microbiota from insect guts, etc. And nowadays, in the age of NGS, next-gen sequencing, most of these strain collections have been sequenced. And so by now, Bayer has a curated database of about 100,000 or more than 100,000 wholly sequenced microbial genomes, and also lots and lots of metagenomes. And as I mentioned at the beginning of the talk, CRISPR systems usually come from bacteria and are very diversified. And therefore, we had reason to hope that our internal databases also contain many different CRISPR systems, and ideally CRISPR systems that haven't yet been discovered in nature. And so, as I mentioned, in 2020, we started to develop our internal CRISPR portfolio by looking in these microbial databases that we've been building up for some time. And so the process we did was as follows. So essentially, there were three components: an in silico component, an in vitro component, and then a more in cellular component. So we first did a bioinformatics screen of the metagenomes in our collection. Found about 10,000 distinct CRISPR systems. Then we did a curation of these systems to essentially narrow the selection down to systems that hadn't been described in the literature, and that we thought had a good chance of working. And then did in vitro screening. So in the wet lab, in vitro screening of about 115 nuclease candidates. Of these, about 30 worked relatively well in the in vitro screen, and then we moved to testing in mammalian cells. And out of this mammalian cell testing workflow, we found two lead nucleases that had really great initial activity levels, and that are now our lead nucleases. And before I finish up my talk, I'd like to share just two slides about these the internal nucleases that we have. So the two nucleases that we found both of them are a lot smaller than essentially the SpikeCas9 benchmark. SpikeCas9 is about 1,400 amino acids. And then our internal nucleases are, one is about 1,100 and the other one is about 500, and again, this is not, there's also in the literature you can find nucleases that are this small, but it's not often that you find well-performing nucleases that are this compact, or also that are sort of around in the 1,100 amino acid range. And so this size reduction could offer advantages for delivery and dosing. The best nuclease that we found is quickly shown here, which is an 1,100 amino acid protein, as it was already shown here. Prefers AT-rich loci, which is also different than SpikeCas9. Can be expressed really easily and stable, and most importantly, shows off-the-bat activity against most targets tested. And while it has a lot of desired activity for a lot of the targets that we screened, the nucleus is also very specific and doesn't cleave at unintended spots, which is also in the CRISPR field when evaluating a nuclease. One is also worried about whether the nucleus cuts somewhere else, but in this case, lead nucleus 1 doesn't. And we've also shown that these CRISPR 2.0 derivatives are possible based on this nuclease. Now, last slide. We're now beginning to see how this nuclease 1 can be enabled for therapeutic applications. One therapeutic application that we're looking at is transgene insertion at a safe harbor locus. And the approach that we're doing here, or examining here, is packaging the nuclease as mRNA into a lipid nanoparticle, and then taking the transgene that we want to express, which here is examples with the reporter gene, into an AAV. Both enter the cell. The nuclease will cut the safe harbor locus after a native promoter. And then with a certain efficiency, the transgene will get integrated into the safe harbor locus, and the transgene will get expressed. And when benchmarking our own nuclease against SPICAS9, which is sort of the industry benchmark, we saw that both cleavage levels were a lot higher at the doses that we used than with SPICAS9, but also the transgene expression also was a lot higher based on the safe harbor locus that was targeted by the nuclease. And so we're relatively happy about now having this toolkit, and let's see where it leads us. Okay, that's essentially the summary of what I just presented. I think so. Essentially, I think did a bit of advertising for gene editing. It's also important to mention that there's still lots of challenges remaining, all right? But I can't mention all the challenges in the 15-minute introduction. But I'd briefly like to mention them on the last slide. So I'd say probably the biggest challenge is still the delivery to the different tissues. Again, liver is easy. Cardiovascular is becoming possible, as Simon will show. Then I think it's not always guaranteed that you will get reliably high editing efficiency in the cell types that you're targeting, and that's especially true for these CRISPR 2.0 approaches. And of course, Simon might also mention is that in the genome editing space, patient subpopulations are usually relatively small. And so to really enable genome editing therapies for essentially the mainstream, there might need to be some regulatory changes. And actually these regulatory changes have begun to happen with, for example, the FDA's recently published platform strategy. Okay, with that, again, like everything in life, this was a team effort. So we have a team of about 15 to 20 people at Bayer working on gene editing. And some of the results presented here were also in collaboration with people at partner companies that I'm grateful for. And with that, I will hand over to Simon. And thank you for your time.
Yeah, thank you very much, Florian, for this first very exciting overview of the work of Bayer. And also thank you very much to the Bayer Foundation for inviting me to this lecture. So my name is Simon Lebek, and as Florian, I was asked for a brief overview of myself. So I went to med school in Regensburg, Germany, where I did my doctorate and my habilitation. And during this time in Lars Meyer's lab, I was working on identifying therapeutic targets for common cardiovascular diseases. So we found several promising mechanisms and therapeutic targets, but we didn't have the means and measures to actually modify them for a therapy. And therefore, I decided to move to Dallas for a postdoc in Eric Olson's lab, where I was working in molecular biology and digged into precise gene editing based on CRISPR technology. 3 years ago, I moved back to Germany, also in Regensburg, where I started my own group, which you're seeing here, where I was combining my previous research interests to precise gene editing to fight cardiovascular diseases. And why is it important to fight cardiovascular diseases? Well, CVD are the most common cause of death in our society, which you're seeing here. And this highlights the need for new therapeutic strategies. Obviously, gene editing lends itself for hereditary diseases. So here I would like to show you 3 major heart diseases that you can inherit. We distinguish them: dilated cardiomyopathy, hypertrophic or arrhythmogenic cardiomyopathy. Taking all these different forms together, they affect 1 in 200 people, and the therapies are limited, aside from heart transplantation. These forms of cardiomyopathies are usually caused by a single mutation, which lends themselves for gene editing correction strategies. So a colleague of mine in the Olson lab was working on an MYH7 mutation. It was an arginine to glutamine mutation, which causes a very severe form of hypertrophic cardiomyopathy. And in patients, this mutation leads to a death by, in 50% of the patients, to a sudden cardiac death at the age of 40 years. So Andreas Chai developed a gene editing correction strategy with adenine base editing to correct this glutamine back to an arginine. And he did that in a mouse model harboring this mutation, where he found that this adenine base editing correction strategy reversed the phenotype. So the treated mice showed a normal heart rate and were also protected from fibrosis. So are hereditary cardiomyopathies now cured? Well, I'm afraid it's not that simple. Here you're seeing an analysis of 209 patients with hereditary cardiomyopathies. And as you can see, there are many different types of genes. And also in the MYH7 gene, which is the most common gene for hereditary cardiomyopathies, there are over 30 different variants in these 209 patients. Overall, there have been more than 1,400 known mutations causing hypertrophic cardiomyopathy, and it's mandatory to mention that each specific mutation requires a specific correction strategy, which complicates a pathway toward clinical translation. Another aspect that already Florian mentioned is that only 5% of the cardiovascular diseases are in fact caused by mutations. So can gene editing also be used to correct common pathomechanisms? And with that, I would like to show you this schematic overview of a cell. We all know in our human body there are hundreds of proteins integral for a healthy homeostatic state. But when the same protein gets dysregulated, it causes disease or various forms of diseases. And this concept was, for example, used by Verve Therapeutics that developed an adenine base editing strategy to ablate PCSK9. PCSK9 is a hallmark for the LDL cholesterol pathway, and Verve Therapeutics thinks to ablate PCSK9 permanently in the liver as a treatment of atherosclerotic cardiovascular disease or also familial hypercholesterolemia. So they developed an adenine base editing strategy to ablate the splicing donor site with a single intravenous injection using ABE 8.8. They achieved a knockout of PCSK9 in the liver. The gene editing components were delivered with lipid nanoparticles. So Verve Therapeutics is very advanced with this liver editing strategy. They already tested it in patients and observed a remarkable editing efficacy and a remarkable reduction in the LDL cholesterol level, which is also really clinically relevant. So while we are excited to further follow up on this editing strategy in the liver, we started other gene editing strategies tailored to cardiovascular diseases, to cardiac diseases, and also looking on pathways that are observed in many different patients and are not restricted to hereditary mutations. So when coming back to this scheme of this mechanistic scheme, one important player in the heart is CAM kinase 2 delta. CAMK2 delta is a central regulator of myocardial function and energetic. And while this can be, when chronically overactivated, it turns into a bad guy and causes diseases. So a couple of years ago, we collaborated with Gilead Sciences and worked on this ATP competitive CAMK2 delta inhibitor GS680 and tested it in human myocardium. It was highly efficient in preventing arrhythmias and also in improving the cardiac calcium homeostasis, but still, this component didn't achieve the translation into the clinic. And this is because current ATP competitive compounds face several limitations and challenges. First, there's a challenge of a substrate and isoform specificity. There are 4 different isoforms that are expressed ubiquitously in the human body. And while the delta isoform impacts the cardiac function, The alpha or beta isoform are central for neurons and brain function. But also the delta isoform is expressed in other tissues. For example, the delta isoform has been linked to learning and memory process in the brain. And this means that an unspecific or global CAMK2 inhibition can cause potentially adverse side effects. They often face a poor bioavailability, but even the best CAMK2 delta inhibitor would mean an extra pill for the patient. And I'm seeing it every day in the clinical routine. My patients have a huge list of different pills they need to take, and clinical data has demonstrated that already with 3 pills per day, up to 30% of the patients don't take their medication as prescribed. When the patients don't take their medication, they're not treated successfully. So our goal is to overcome these limitations by precisely and permanently modulating the CAMK2 delta pathway. And we think that this can be achieved with CRISPR-Cas gene editing to introduce a protective mutation and disrupt the pathogenic signaling cascade. Here you can see an overview of the structure of CAMK2 delta. We have 3 main domains. And within the regulatory domain, there are 2 methionines adjacent to each other. These methionines can be oxidized upon oxidative stress, for example, upon ischemia-reperfusion injury. And once oxidized, they increase CAMP kinase activation, leading to cardiac malfunction. We designed a CRISPR-Cas gene editing strategy to precisely modify these adenines into guanines on the DNA level using adenine base editing technology. The resulting GTG codons were then recognized as valines by the cell machinery and rendered CAMK2 delta resistant to oxidation. We packaged this editing construct into an AAV, and as Florian mentioned, AAVs have a limited packaging capacity, so we needed to split the editing construct. We have an N-terminus carrying the ABE8E base editor and the N-terminal spry nickase. In the other virus, we had the rest of the spry nickase. Thanks to these both N-inteins, the split virus reassembles to a functional adenine base editor in vivo and 2 copies of the single guide RNA. Use of a cardiac troponin T promoter enabled or ensured that the base editor is exclusively expressed in cardiomyocytes and nowhere else. Here I would like to introduce you to our experimental design. We used very normal C57 black 6 mice, and at the age of 12 weeks, we subjected them to ischemia-reperfusion injury. We then injected the editing components directly to the heart and followed up with a series of echocardiographies and further molecular analysis. Here you can see a healthy sham control mouse, echo recording of the left ventricle. And as you can see, the anterior and the posterior wall are getting quite close to each other, indicating a healthy cardiac function. As expected after ischemia-reperfusion injury, with or without a control virus, we found a left ventricular dilation and also an impaired contractility. So I hope you can appreciate that these anterior and the posterior wall, they are not getting quite close to each other anymore. And this was substantially improved in mice that received a CAMK2 delta system. We then harvested the tissue after the injury and analyzed the editing efficacy. In fact, we achieved a more than 80% editing efficiency at the anterior wall of the heart, and this was this region where components were injected. As I mentioned earlier during the talk, there are various isoforms of CAMK2 delta, which means it is important to edit to achieve a high isoform specificity. Therefore, we performed deep amplicon sequencing, and we didn't observe any significant editing for the alpha, beta, or gamma isoform. It was all below the threshold of 0.2%, and comparable to mice that were treated with a non-editing control AAV. Another issue or another challenge is achieving a tissue specificity. So we performed off-organ analysis to test whether our troponin T promoter was in fact cardiomyocyte specific. And indeed, we didn't observe any significant editing of any CAMK2 isoform in the brain, the skeletal muscle, or the liver, demonstrating us that our editing approach was not only isoform specific, but also tissue specific. As ischemic perfusion injury and heart attack is frequently linked to fibrosis or scar formation, we performed trichrome staining of mouse hearts, which you can see here. In blue is the fibrotic tissue, and I hope you can appreciate that in the heart attack mice, there is significant fibrosis or even scar formation. In edited mice, it was not completely absent, but at least strongly attenuated, and we found a substantial reduction in fibrosis, which was consistent in replicates and significant across the groups. Moving toward clinical translation, we decided to generate a humanized CAMK2 mouse model, which is an important step as the gene editing strategies are based on the DNA, which is different between humans and mice. So using CRISPR-Cas homology-directed repair, we replaced the regulatory domain of CAMK2 delta with the corresponding human sequence to generate a humanized CAMK2 delta mouse model. And this mouse model allowed us to deploy the gene editing strategy optimized for the human genome in vivo in mice. And with this optimized human mouse model, we were able to replicate our previous findings. So we found a recovery in cardiac function after a heart attack. We also put the mice onto a treadmill to examine the exercise capacity and found a recovery on the treadmill and a higher maximal velocity. Both parameters correlated very well with each other. So the better the heart function, the better the performance on the treadmill, which was an internal validation for our strategy. Another key feature of heart failure is the occurrence of arrhythmias. So we performed right heart catheterization to test for arrhythmias. Here you can see an ECG recording of a healthy sham control mouse. And immediately after the burst stimulation, there's a just normal sinus rhythm. In a mouse with a cardiomyopathy, we've detected severe arrhythmias, which were completely absent and protected in mice that received an AAV editing construct. Of course, the major question is, could this in fact work in patients with heart disease? And therefore, we teamed up with the cardiac surgeons in our department in Regensburg, and we are now obtaining myocardial biopsies that we are slicing and are able to cultivate for several weeks and months to actually measure the contractility and the occurrence of arrhythmias in treated and untreated human patients' myocardium. And I hope I can show you some data which we are producing or which we are testing right now. And with that, I would like to conclude. I hope I could demonstrate that also common human diseases can be targeted by silencing pathological signaling cascades with CRISPR-Cas gene editing. After a careful design, we are able to edit permanently one specific target, meaning one specific gene. And with a tissue-specific promoter, we achieve editing exclusively in one organ. And I think that with this approach, we may achieve sustained therapeutic benefits with potentially less adverse side effects. And now at the end of my talk, here you can see my current group and my funders. And thank you very much for your attention. And I'm happy to take any questions. Thank you very much.